great pleasure to be here to speak about a very important topic, cell and gene therapies and the value cell and gene therapies bring to patients, but also to take the perspective from the healthcare systems. And I think it's really a very important discussion which we're having here today. We need to bear in mind that almost all cell and gene therapies are one-time treatments. And I think this is really important for the discussion today. And so there are no long-term costs as you have them in chronic treatment options. I think it's really important that we bring that into the right perspective today. Also, I wanted to mention that many cell and gene therapy companies and products are generating real-world evidence, working with registries to really meet the demand of the payers and the healthcare systems. And also what we have seen are many outcome-based agreements, certainly something which we will discuss later on. Thank you for being with us.
Many thanks. I am privileged to present you my understanding of how gene therapy is really coming very quickly, certainly in my clinics. And I will take you through a few examples. I work as a pediatric neurology in London, and I see children with mostly neuromuscular disorders. Next slide, please. These are my disclosures. As you can expect, I'm involved in a number of clinical trials, also provide some advice to companies. I'm not going to be remunerated for this particular presentation. This is part of my academic talk. Next slide, please. So the first high-level question is, what are genetic diseases? And you may or may not be aware that there are approximately 10,000 rare disorders that affect 6% of the population in Western society. So on one hand, the majority of us doesn't have these rare disorders, but a large number of the population does. 80% at least of these 10,000 rare disorders have a genetic component. They are often disabling and expensive to manage. Most of these conditions are inherited from one of our parents. And 1/3 of the children with rare diseases die before the 5th birthday. This is one area of my work and expertise, next part. And this is, on the right-hand side, an example of how the conditions can be inherited from, in this particular example, from one affected male, in gray, to his offspring, and how this then continues to be transmitted. There are different modes of inheritance, but this is beyond the scope of this presentation. Next slide, please. So we have more than 20,000 genes distributed in the 23 chromosome. On the left-hand side, you see the 23 chromosome. Each of us and every single cell in our body has one copy of the chromosome from our father and one from our mother. And therefore, every cell contains, every chromosome contains thousands of genes, and occasionally some of these genes may have a misspelling, and that is what causes a genetic disease. Next slide, please. Now, how can you correct permanently a genetic disease, and how you can do it? Now, traditionally, in the more distant past, although this is still used, organ transplant was the only way to deal with some genetic disorder, and I will give you one or two examples next. In the last maybe 15 years, cell-based delivery.
Yeah, you're a big shit gobbler.
Somebody muted me, but the cell-based delivery of corrected cells have been also used, and I will provide you some example. Next part. But I think what is really happening at a faster pace is that the direct delivery of a transgene or genes that have been corrected to patient directly. Next slide, please. So in terms of the organ transplant, this is something probably many of you are aware of. There are a few conditions where if you have a genetic disorder, an example is thalassemia. In thalassemia, the problem is in the ability of your bone marrow to produce normal red cell. In that case, you can do a transplant of the bone marrow and take a donor, give the bone marrow to the children. Cell-based delivery of corrected cell is a relatively more recent modification. You can take, and these are, for example, conditions that have been really completely transformed are immunodeficiency. You may remember several decades ago, the bubble boy syndrome. These are children who cannot be exposed to the environment because of the immune deficiency. And next part, you can take some of the cells from these affected children, from the bone marrow, take them, you culture them in the laboratory, then you take a virus, you infect the cells with this particular virus that has a corrected genes, next part. And then once you expand these cells in the laboratories, and there are enough, and the gene is inserted where you want, you can put the cells back into the individuals, and that individual will be cured for life. This has been transformative. However, you cannot do this for vast tissues. For example, you cannot do this for muscle, you cannot do this for complex tissues. Next slide. And for some of these, other more complex conditions, for example, if you wanted to correct all cells or many cells in the brain or in your muscles and so on, there are different strategies. And this is the direct delivery to the patient of a gene using a vector. This is also a virus, but this time the strategy is different. The first, you take the virus and you strip from inside all the genetic material of that virus. You do this for two reasons. Firstly, because in that way, the virus is even safer because it does what you want, not what the virus would do, next part, but also because you need to make space for the gene you want to reintroduce so that can be used for the patient. Then you use this virus really as an envelope to deliver to the patient the gene you want. And this is often done by intravenous injection. And as we was mentioning earlier on, this is a one in a lifetime injection, or in some condition, directly in the organ of interest. Next part. And these are examples of both approved product on your left-hand side with some cancer, some eye disease, some metabolic disease, and spinal muscular atrophy, where in Europe now there are approved therapies. And on the right-hand side, people like me are involved in multiple clinical trials for a number of neuromuscular condition and metabolic condition. Next part. And I think this is a very recent review in one of the Nature journal regarding how this particular type of gene therapy is really gathering momentum with an increased number of clinical trials that eventually and hopefully will come to approved products to be used in clinics. Next part. And these are just two examples, well, actually, it's one example of one particular family I've been involved with. These videos have been consented to be shared. And you will see on the left-hand side first a girl who has a wheelchair and she's carrying her sister in front. Now the girl on the left-hand side, she never walked. She's been using the wheelchair. She needs to be ventilated during the night. And you can see the sister that has been treated very early on. She clearly is a very different child. And today, at the age of 3 years, I cannot tell the difference between this child and a normal child. And therefore, that gives you a very tangible example of what gene therapy can do in the real world. Next slide. So I think that if you like, this session is also been organized to make you think how the future would look like. I think that there is no question that the number of clinical trials and products under development for gene therapy is expanding. Of course, these are still a rare condition, so it's not that there will be thousands of products coming your way in the coming days. But at the same time, for the first time, some of these gene therapy products really have the possibility to shape the way we do our clinics, the way the healthcare system in European countries needs to be organized. We need training, we need registries to collect the long-term effect of this medicinal product. And of course, clinical services need to think how to reconfigure themselves to deal with how different patients will be in the future. And the next and final slide is firstly to thank the people who work with me in my hospital, at the Downing Street Hospital, but I think really also to thank you because on the right-hand side, you have the European project. I'm at the moment coordinate the BIND European project, but I've been involved in many projects related to rare disorders, and I really thank the European Commission because they work that we and others do on the rare disorder has received a major help from the European Commission. Thank you.
So first of all, just thank you for the opportunity today to present on cell and gene therapy manufacturing. I'm going to begin with a high-level overview of comparing the traditional drug manufacturing process to the autologous CAR-T cell therapies. So, you know, a lot of people would be familiar with the traditional manufacturing process in terms of synthetic drug manufacture, tableting, for example. This process is a very fast process in terms of manufacturing. It takes days to complete. The testing occurs at defined steps in the process. The product tends to be very stable because it's synthetically made. It has a long shelf life. It's typically stored at ambient conditions at room temperature. And it's quite simple in terms of supply chain in that you can hold inventory at different parts of the process to help with demand for the customer. Just looking at autologous manufacturing or CAR-T therapy, it has longer processing times. The patient has specific therapy, so it's customized to the patient cells, so very much individualized. The testing is continuous, so it's not at defined steps in the process, it's right throughout the process. It's time critical to treatment because of the urgency of the disease. The drug tends to be frozen storage, which adds complexity to the manufacturing process and supply. And it's micromanaged, so right through from when the patient is identified through to when they're actually treated. So that it's fairly heavy in terms of manual interventions. Just moving on to the next slide. So this slide is giving a high-level overview of the manufacturing process for gene therapy in particular. So starting with one vial containing approximately 10 million cells for approximately 3 weeks, they're supplied with fresh nutrients so they can multiply and expand in number. From there, then the cells reach up to 1 billion in number and they're transferred to a bioreactor for continuous expansion. The plasmid DNA is added to instruct the cells to produce viral vectors and that contain the therapeutic replacement gene. A few days later, the cells are broken open and the full gene therapy vectors, empty vectors and other components are released. So this part, steps 1 to 3 are the upstream, or what we refer to as the upstream part of the process. And then steps 4 and 5 are on the downstream side. So step 4 is now getting into the stage where we're filtering the product to make sure that it's pure and safe for the patient. So any empty vectors that lack any therapeutic replacement gene is eliminated from the process. And then the final stage is the aseptic filling of the product into vials. In terms of Zolgensma, the gene therapy Zolgensma, each order for per patient is based on their weight. So it's not as specific as CAR T in terms of their cells used in the manufacturing process, but it is weight-based. So there is an element of customization of the product. So the key points, sorry if we just stay on that slide, please. The key points are, in terms of gene therapy, they're small batch sizes. It's quite complex and difficult to scale up. The manufacturing process is 24/7 monitoring, and this gives us a surety of the product safety, purity, potency, etc., which is tested throughout the process. It has short shelf life, so 12 months shelf life. Typically for traditional products, it's 24 to 36 months. And again, there's complexity with how the product is stored. So in terms of Zolgensma, it's minus 60 degrees Celsius. Again, adding complexity in terms of the distribution piece to the patient. Next slide, please. So this is my final slide, and it just gives you a snapshot of the timeline from manufacturing through to treatment of the patient for Zolgensma. As mentioned previously, there's a 12-month shelf life on Zolgensma. 5 months of that time frame is used for the manufacturing, testing, and QP release of the product, again ensuring the efficacy and safety of the drug for the patient. Once the drug is released, then the supply chain speeds up because of the urgency of the patient and the criticality of their disease. We can't hold inventory because of the short shelf life, and because we have to do weight-specific dosing. So we then manufacture the secondary packaging into regulatory packs, with their specific EU requirements in terms of blue box labeling, EU FMD regulations, etc. And so that packaging happens within 48 hours along with the QP release. And then it's shipped out to the site of care between 24 and 48 hours, depending on the location of the hospital within Europe. So just to conclude, and summarize, the manufacturing of cell and gene therapies are quite complex in comparison to traditional methods. They target orphan drugs, so it's small patient populations, and then the time criticality is a fairly big factor due to the disease and the urgency to treat the patients. Thank you.
Thank you very much for inviting. Thank you to the originators of this initiative and Alex for your introductions and setting the discussion in a right framework. I lead currently in Novartis healthcare system engagement and capability building, but only a couple of months ago, I was in Novartis Gene Therapies responsible for gene therapy public policy. So what I would like to share with you is my perspective from the Novartis Gene Therapies as a public the policy the lead on this topic. We will have a discussion about focused on the value of gene therapies and how healthcare systems are ready to embrace this value and make sure it materializes for the patients and for healthcare systems. I'm glad that you already had first session where you had an opportunity to go to the science of gene therapies, how it works and how it is manufactured. So I will not be focusing too much on that, assuming that some of the members of the audience today also attended the previous session. So if it's not the case, and if there are some questions about that, I'm happy to also address this later in the question and answer session. Next slide, please. So really, arrival of gene therapies marks the change of the new era in medicine. This is really a pivotal point where we have a great opportunity today to treat some of the devastating rare diseases with huge burden on patients and societies. But also with that comes a hope that in the future, as science advances, the gene therapies could help us to address the burden caused from some of the common diseases, which are traditionally currently treated with more traditional type of therapy. So that's an important thing to bear in mind. That really we are at the pivotal stage now in the history of medicine. Gene therapies have a bunch of different characteristics that our healthcare systems have not really encountered before, and therefore they are not necessarily ready. So if you want to say this is a 21st century medicine and technology that we need to make it work for 20th century healthcare systems. The first of all, if we look at the mode of administration, the conventional therapies, which most of them have been designed for chronic conditions, they require repeated treatment, repeated administration for months and for years or for the length of the patient life. While gene therapies usually are a single dose and short course administration therapies. That has a big value for patients and for healthcare systems as well. And the other important dimension of the difference here is the mode of action. Most of the traditional therapies for chronic condition, they address the root, not the root causes of the disease, but their symptoms, and they try to reduce the symptomatic burden on the patient. While the gene therapies, they treat the diseases at the root cause with the potential to halt the disease progression or even provide the curative outcome for the patients. Duration of the treatment is another important difference between the conventional gene therapies. Conventional treatments must continue in order for them to exert the effect. So sustained effect is only possible if you continue treatment with this therapies like cholesterol lowering therapies or the cardiovascular hypertension management therapies and etc. While the gene therapies, it's a basically one-time intervention with multi-year benefit, potential for lifelong benefit, and that's extremely valuable for patients, especially the patients with the type of diseases that gene therapies are currently designed for. When it comes to access, conventional therapies really shape the way the current value and economic assessment works. That is usually the way the patient gets access to this therapies. The value assessment processes that are established, they are really tailored at the type of evidence that are usually generated from RCTs, very robust evidence on the effectiveness from the beginning. Well, gene therapies are challenging that, bring greater uncertainty in long-term outcomes, and novel study designs are not always easily managed by the established value assessment mechanisms. And then when it comes to the payment, the traditional therapies are because they have to be taken for the course of their life, and the systems that we have currently to pay for those are designed for this type of therapies, while the gene therapies, which are the single administration, they require upfront payment for the whole value of it, and that's also makes it completely different animal, if I may say, from the conventional therapies. The next one. Very briefly to touch and refresh perhaps the discussion that you had during the first session. Then when we are talking about gene therapies, we are basically talking about 4 modalities through which gene therapies can exert influence. We're talking about the gene replacement, when the function of a dysfunctional or missing gene is replaced with the functional copy of a gene. This very often designed for monogenic diseases. Gene addition, when additional therapeutic gene is introduced that targets specific elements, mechanism, and disease pathway. Gene inhibition, we mean by gene inhibition silencing the expression of a mutated gene. And then gene editing, editing the gene, which means the patient genome is edited to correct the specific mutation that causes a disease. We don't go into the kind of detailed science of that, but I just wanted to put it out there as a kind of a definition of the four major modalities of gene therapies. Currently, most of the gene therapies are for monogenic rare diseases, but we see their potential also in some the other diseases, but science is not yet there. Whether we will get there successfully or not certainly will depend on how the markets and healthcare systems embrace the current early birds of gene therapies, and what kind of incentives the innovators and developers will see in pursuing the relentless search for application of gene therapies into other more public health related issues. As I said, we are not yet there, but we might be there if we create the right pathways for gene therapies to deliver the value to patients and health systems, and the incentives is there for the innovators. Next slide, please. As I said, let's switch gears and talk a little bit more on the value of gene therapies. Gene therapies generate a high expectation from the society. We expect them to be very effective than some of the traditional therapies. Particularly the efficacy of the treatment, the effectiveness of the treatment is very much determined by the time of the initiation of the treatment. And many of diseases, as we know already from the previous section, are genetic diseases. They have a certain manifestation time, certain lag time. Earlier we initiate the gene therapies, particularly at presymptomatic stage, the greater the chances of the efficacy. A clear example of it is the spinal muscular atrophy, when by the time that the children are diagnosed, maybe 90% of the neurons are already dead. So if we could do that at an early stage, we certainly would have a much higher chances of preventing the lifelong disability. Society also expects from gene therapies favorable balance of efficacy and safety. There is the expectation that we know this is going to work. There are certainly some possible side effects of the therapy, but overall, the promise of the benefit is greater than the risk. So this is also the perspective that regulators embrace. There is an expectation of durable response. It's hard to have that evidence at the time of the approval of the medicine, but what we know about the physiology, biology that underpins the science of gene therapies, it is highly likely that they have a durable and sometimes the lifelong impact. And a targeted treatment, this is really an epitome of the personalized medicine, if you will, because this is specifically targeting the root cause of the disease. So all these factors would make the gene therapy something that is most likely to deliver very high value for the patients. And if you look at some of the studies that have done recently, the average quality of life gained from gene therapies, it's almost 10 times higher than the quality that are gained from the conventional therapies. That's an important thing to keep in mind. So that's as far as the patient value is concerned. So superior clinical efficacy and probability of giving the lifelong benefit. Let's look at the healthcare system value. Next slide, please. The economic impact of gene therapies, and which is particularly important for healthcare system, depends on what aspect of the patient value they are mostly enhancing. So we have on the one side the length of the life. There are some gene therapies that predominantly might work on extending the length of the life. And in this case, the gene therapies are generating the substantial increases. But if that length of the life is extended without a significant increase in patients' quality of life, and without significant increase in the decrease in the probability that patients will have to continue using the other supportive healthcare, probably there is not so much of economic gain from that, but there is a huge patient gain from this therapy. But there are the gene therapies that can actually extend significantly the quality of life patients, make patient pretty much symptom-free, and enable patient to return back to the normal life. And this is the huge economic gain for the health system and for the society. Usually there are no such strict boundaries between the types of gene therapies. This is basically, I put here for helping you to understand in which directions this patient value can finally materialize into the economic value. Gene therapies that extend both life of the patient and also quality of life of the patient are the ones that have tremendous value for patients and very big economic value for the healthcare systems. So let's look at this a little bit more. Next slide, please. On this slide, you don't have to read everything on this one. What I will point out, which kind of visually, what should come quite explicitly from what you see here, is the cost of the treatment of various gene therapies that already exist relative to the cost of treatment from other non-gene therapy alternatives for those diseases. And the orange boxes show you at what point the price of the gene therapies will equate the cost of the alternative treatment for this disease. And as you can see, pretty much all those therapies that are put on this chart, between 5 to 9 years, the cost of the one-time cost of that will kind of balance the treatment cost of of these diseases through traditional therapy. So if we think that for whatever the gene therapy you might pick from this list, these are the, let's say, most expensive therapies. Actually, the other side of that, they might be not so much of expensive therapies, they might be high price therapies, but they might end up being actually the most cost effective and least expensive way of managing that patient over the course of life by the healthcare systems. So that's kind of the other side of the coin, high price, but over the lifelong course of the patient, they could be actually the most cost-effective, efficient way of treating them. So as you can see, the healthcare systems are certainly gaining a huge benefit after 5 to 9 years from that perspective with gene therapies versus the traditional treatments. Next slide. We are also asked very often the question, and this was something that researchers themselves got very interested, what is going to happen to healthcare systems if the current pipeline of gene therapy research and development pipeline continues as it is? And this is a study that was done by Wong and colleagues in the US. We could also extrapolate it for Europe, but I just wanted to show you whatever was in the paper and the evidence rather than my personal extrapolation. The bottom line here is that gene therapies are not going to bust healthcare system budgets. They will not bankrupt the budgets, and the budget impact from those would be relatively small. If we consider into account and take into account that all those clinical programs that are currently in the pipeline will end up successful, by 2034, we will have about 306 billion total spending over the 15 years on gene therapies. On annual basis, about 20.4 billion in the United States. But if you consider that United States, by the time will be spending about 6 trillion on healthcare. So just if you take a single year as an example, let's say 2028. In 2028, based on this projection, we expect that the projected tax revenues from all the gene therapy related activities in the US would be, the cost of the gene therapies relative to the tax revenues would be only 0.44%. And projected, as a percent of the healthcare spending, only 0.39%. So the question to you, do you consider the spending on gene therapies, which has, as I mentioned earlier, quite a big health gain potential for the society, especially in 14 years from now, 0.33% as something that is challenging healthcare system sustainability and the budget impact. And the picture looks most likely the same for Europe. That's highly unlikely that in, let's say, 15 years from now, that we will be spending with more than 0.4% of our budgets, healthcare budgets on gene therapies, and consider the value that they can bring for that. So the clear answer to that, based on this projection, is that it's not going to challenge our healthcare sustainability. And these are the very I would say, over projections, because not all those clinical programs will probably materialize, and we expect there should be price challenges as well. So the list price that went into the assumption of this based on the early examples of gene therapies will not be the same, let's say, in 10 and 15 years' time. Next one. So let's talk about the, in the next 2-3 slides, what are the current big challenges the gene therapies currently face in healthcare systems. I mean, traditional models, when we look at the therapies, basically have the 3 elements to that. First is the clinical inquiry. Is this product working or not? And here, the gene therapy, when it comes to the gene therapies, there is a challenge. Not only on the economic side of it. Because if we look at the HTA agencies versus regulators, they have slightly different perspective, right? Regulators are looking at the benefit and the risk, while the HTA agencies are more interested in relative effectiveness and the value for money. And the gene therapies, which have inherent uncertainties in the data at the time of this assessment, are somewhat disadvantaged here if we don't change the paradigms that are currently used in the economic, the clinical, and also the economic appraisal. We don't have a lot of comparative data at the time of the bringing this therapy to the appraisal stage. We don't have comparative assessment to the other the therapies. Usually they are not based on the RCTs. So looking at it from the traditional lenses would certainly put them at a disadvantage. The same goes for the economic appraisal, right? Because price is usually negotiated upfront, and the key considerations that are driving the economic impact is the budget impact, short-term the cost impact for the payer, and benefits to the families and wider societies are not always considered. And that's again puts the gene therapy in relatively incompetitive position compared to the traditional ones. The short-term view is at odds with this long-term potential benefits and potential curative benefits that with one administration lasts for the life of the patient. And then financial dimension also very important, because our healthcare systems are used to pay continuously, like a rent, for the therapies. As long as the patient therapists are taking therapy, you pay for that. You stop the therapy, and the value stops there for the patient, and then you stop payment as well. Well, in gene therapies, all the value is actually at one time is transferred to the patient and health system, and the payments are, the price reflects that one-time value. So how we can manage that, which is not so difficult, to be honest, to fix that discordance between the current financial mechanism and the way the gene therapy pricing is constructed. So these are the 3 big challenges. So next one. So what we need to do in order to fix this is basically look at the 3 things. Make sure that we have a much greater flexibility in the evidence requirements. We should be much more open to non-traditional trial design. We have to accept some evidence gaps and mitigate them later on with perhaps the payment mechanism. And the other big thing is to have a much broader value assessment frameworks to consider the patient's broader value, societal value, and not only focus on the traditional cost effectiveness models that we have been using very often in relation to the traditional ones. And then innovative payment models are able to resolve many of those uncertainty issues as well as affordability issues. If we could mimic the traditional payment mechanisms in relation to gene therapies through a deferred payment mechanisms, the installment payments, we could also incorporate some of the outcome dimensions you need to account for the potential uncertainties for the evidence. Those things could be addressed. So the question here becomes, Are we ready to make these changes? They are not extremely difficult. They require just the political will and the collaboration. And from our side, as far as Novartis is concerned, I'm sure the same would go for the other industry players. We are really motivated and eager to play with the stakeholders together to come up with the solutions that works for the patients and works for the health systems and also works for us. So given that we need to have some more time for the question and answers, I'll stop my short presentation here and hand it over to the facilitators. Thank you very much.
So if we just move to the next slide. My name is Avril Daly. I'm the CEO of Retina International, and that's a patient-led global organization. We have members all over the world, including in Europe, who are themselves organizations who've been funding and supporting research into retinal degenerations for over 50 years now, particularly in the area of gene and cell therapy. So as you can imagine, we would have started working on gene therapy, in the 1970s when very little was known. To get to this stage now is a critical time and a very hopeful time for us, but it's not without challenges. So really what I wanted to talk to you about today was, just the basics really of genetic disease and what the challenges are for us on the ground and how we need to look at how we assess value and also what policy actions can support us in our efforts. So as you will all know, in Europe, a rare disease constitutes fewer than 1 in 2,000 citizens. And some will say there are about 6,000 rare diseases. Others would say there are about 10,000 rare diseases. Of course, they're being described every day. But we understand that there are 30 million people in Europe who are affected by a rare disease. We must recognize that within the context of that, about 80% of rare diseases are genetic in origin. So we must understand that that's a huge portion, and that the onset generally occurs in childhood, with about 50% of rare diseases, causing mortality, unfortunately, in children. So the main groups that you would be familiar with in the context of rare disease are the metabolic, neuromuscular, I should say, autoimmune. We have developmental anomalies, bleeding disorders, cardiovascular, respiratory disease, skin disease, rare cancers, and also sensory diseases, which my organization would be involved with. But it's very important to consider, and it's already been mentioned this morning, that we're learning more and more about general diseases or common diseases, and that some of them actually are inherited and have that genetic component. So it's really looking into the future. It's much broader than just the rare disease space. So next slide, please. So how do we address the challenges in Europe through gene therapy? So gene and cell therapy has huge potential, as we've already said, to treat inherited and complex conditions and to change the lives of a very significant number of people with a very challenging unmet need, and to reduce the burden of disease on those patients and carers. A survey from a Rare Barometer study survey from Novartis a couple of years ago showed that people who are caring for patients living with rare diseases and the patients themselves spend about 6 hours a day just on administering therapies and treatments and various different caring for people with the disease. So that's a significant amount of time, as you can imagine. And so if we can improve the delivery of care and treatment with genetic disease, we can really reduce burden. We can reduce disease burden on those that are affected, and the burden of treatment, which can be very complex and can take a lot of time, as I've already said, but also, the actual burden on those HCPs who are delivering the care. And we can also address growing societal inequalities. People who are living with rare diseases are often marginalized and lost within the system, and can, have all of the realities that affect you in those circumstances. And ultimately, we do believe that gene therapy and cell therapies can reduce cost because of their personalized nature. And we've heard a lot about that already. So next slide, please. So the key challenges, as I've already said, from our perspective, these are just key challenges. We've heard about the issues concerning pricing, reimbursement, and access. But from our perspective, it's an infrastructure issue. It's an issue of how we assess value. And it's also an issue of policy action and how we can join the dots on the various policy actions on this issue and what's relevant to this issue. So next slide, please. So a lack of infrastructure to support the delivery of genetic services is a real challenge for us right now. When you receive a genetic diagnosis, it's a fundamental step on your journey as a patient. And we know this. We know there are huge challenges with diagnosis in the rare disease community. We know and are learning more about newborn screening. Genetic diagnosis is something that seems to slip through the cracks a little bit. And it's of course fundamental. If you're talking about delivering a gene therapy, you have to have a genetic test. So we need a best practice model in Europe on access to genetic testing and counseling services. In Retina International, we undertook a study last year to look at the processes of accessing a genetic test and then the patient experiences, which were in fact very different. If you look at it from a process perspective, it seems okay. But if you talk to the patients, then it's a very different reality. So our study found that with 419 respondents from patients living with IRDs, which are inherited retinal degenerations, and this 42% of respondents did not receive a genetic counseling prior to genetic testing, and 33.89% of respondents did not receive genetic counseling after receiving a genetic test. Now, why is that important? You need to understand what a genetic test is and its relevance to your family and its impact on you prior to agreeing to the test. It's a very complex and very important issue. And subsequently, to not have genetic counseling is handed data as a patient that is not interpreted. And it might alarm you, and it should alarm you to know that organizations such as ours receive this data from patients who don't know what to do with it. So you can imagine the challenges that we are facing because we don't have that sort of a support for this specific area. We really need to increase awareness and training within HCPs. We have a global shortage of genetic specialists. We have a global shortage of genetic counselors. We don't recognize genetic counseling as a medical discipline. And there's very little training. It's very sporadic in Europe. It's not very consistent and it's not available everywhere. And that's a challenge if we are to talk about delivery of gene therapy in the healthcare system. You know, it's the burden on the patient currently to navigate the system to find a genetic diagnosis. And really, we need to address this. And also how we assess value. I can only speak from the perspective of our community, which is a small community, a small population, where we're talking about inherited retinal degenerations. So if you're looking at health economics and you're looking at how a patient affects the healthcare system and as a cost to the healthcare system, well, we really don't fall into that group because vision loss is unlike other conditions where, you know, you don't go to see your doctor very often, you're not sick per se. And health systems costs are the lowest cost types when conducting socioeconomic analysis on the impact of vision loss of inherited retinal degenerations. We have done this in Europe and we have done this in the United States and Canada. And what we're learning is that what really affects this population are very high rates of depression, anxiety, mental health issues, huge financial stress. There's impact on access to education, on access to employment, and huge productivity losses. There is impact on the caregivers, and they're often missed in not just the community I represent, but in all rare diseases. The burden on caregivers is enormous. People have to leave work, you know, and they have to navigate a very complex system. And of course, there are many intangible costs that we don't assess when we are looking at the traditional HTA model. And I think this is a very important point that we must look at. We need to look at the person, the patient, and their families, and the entirety of the supports that they need, which may not necessarily be specific to healthcare. And that's important. And of course, this is also something that affects other rare diseases. It's not just our community that have this issue. So next slide, please. So we know that there is a lot being done in Europe, and you know, rare diseases have been a priority, a public health priority in Europe for many years. But the issue of genetics, you know, is something that, although fundamental in rare diseases, we probably don't discuss it enough. There are issues with that. And I think that affects how we act from a policy perspective. But there is a lot of good happening. You know, there's a lot of joined up thinking already. There's a lot of initiatives that can support access, that can really look at the landscape and try to address the gaps and the issues. And, you know, I've listed many things here that are related to to how we enact policy in Europe. What I think is very important, and we should say, is that when we are looking at the policy landscape, we need to consider, is it consistent? Is it aligned? is one element of policy going to have a detrimental effect on the other? And I think this is very important as we look at this really hugely valuable and hugely encouraging and hopeful area of health. You know, sometimes this can be an unintended consequence, and it's something that we in the patient community are keeping a very close eye on. So next slide, please. So as I said, you know, the issue of genetics and you know, its impact on rare diseases and, as I say, other conditions is something that probably we don't speak enough about, and there is a lot of confusion about. So the roles of genetics in healthcare is often very unclear. It can often be considered as niche. Well, it's not niche. It's but it's a model of care. It's a model of delivery of care and personalized care and therapies into the future. It is seen as the future, but it's really now, as we know, and we've already heard, gene therapies are available and there are more to come. And under-resourced genetic services are a real impediment to access here. And we must include testing and counseling, you know, and support them in the development, you know, and their development in the context of the healthcare systems. And we also must consider, you know, how we deal with that data, you know, in the development of registers, you know, is something that, you know, is discussed a lot in this community. We must realize the full potential of innovation in gene and cell therapies. Gene and genomic strategies are needed to support the delivery of care and the alleviation of the care burden on patients, carers, and HCPs. And if you look across Europe, there's huge disparity here. There are some very good examples of gene and genomic strategies, but in other countries, there is no strategy whatsoever. And unfortunately, and in some countries as well in Europe, genetics is not seen as a health discipline in and of itself. So how is it ever going to be catered for? We need to rise all boats. And, you know, to my previous point, we can do that by enacting very robust policy actions in this space. And that's really very important. You know, as I said, we need to be consistent. We need to support the delivery of these therapies. There is a lot to be discussed. We've heard from our previous speaker, you know, on the issue of cost and access and reimbursement. And looking at a system for 21st century therapies is really important, because currently we're applying a 20th century model and a 20th century thought process to how we're going to deal with those. And that's really not going to work for us if we want Europe to be a leader in innovation, not only in the development of these therapies, but in how we deliver them and how we think about them and how we think forward into the new era of gene therapy. It's really important to know that we are talking about a transformation, but sometimes, and I know politicians get a little scared when they hear the word transformation because it might mean a huge amount of cost, but really we have the intellectual capital in Europe. We have a lot of these systems and we are in the process of networking them together. And those networks, like the European reference networks, for example, offer huge hope and they really need to be supported. And if they are supported, it will go a long way to ensuring that we have laid the groundwork for the future. And really, I think that's really what is important. Let's look at what we have, let's support what we have, and let's work together. Thank you.